let's talk about what mutations are. So, mutation is when a DNA gene is damaged or changed in such a way as to alter the genetic message carried by that gene. It can happen either when mistakes in when the DNA is copied, which are also spontaneous mutations, which is mistakes when the DNA replicates itself, or a result of environmental factors, which are induced mutations, which are agents in the environment that causes change in the DNA structure. An example of this that you will learn more in depth is the sickle cell anemia, which is a mutation in the gene that instructs a protein. Causes red blood cells to become abnormal and in a sickle shape, but this can be also a positive mutation. It protects against malaria. So here are the three types of mutations. Point mutations in specific. Substitution, which is specific spaces in a nucleotide are swapped. Insertion, one or more extranucleotides are inserted. And deletion, one or more extranucleotides are deleted. And nucleotides are those random letters that you see in the DNA ladder.
All right, now let's learn about how mutations form. Mutations simply form when DNA makes copying mistakes, so the sequence of the DNA is changed. Another way is through viruses that cause infections, such as HPV. Mutations also formed when you're exposed to ionizing radiations, and you might know some medical procedures that contain these, such as CT scans, X ray, and PET scans. However, that is not to say avoid x-rays because it is necessary for certain procedures. Lastly, exposure to mutagens or agents that cause genetic mutations cause mutations to form. A common example is smoking. Smoking can increase the risk of cancer, which is a type of mutation where cells multiply uncontrollably. Now let's move on to the different types of mutations.
Now let's learn about point mutations, or a change in single nucleotide of DNA. They are the most common type of mutation. That nucleotide that can be switched to another nucleotide, deleted, or a single nucleotide can be inserted into the DNA, causing the DNA to be different from the normal gene sequence. Most commonly, they involve the substitution of one base for another. This image on the slide clearly shows the difference between an original sequence in DNA compared to a point mutation. Point mutations typically don't cause much change in the underlying organism. Now let's move on to substitution.
Next, we'll be talking about substitution mutations. This happens when nucleotides are replaced by a different base pair. So if you look at the image on the right, you can see that TGC has been replaced by GAT. These mutations can result in either no issues or major issues. Next, we'll move on to insertion. An inversion mutation happens when there are two breaks in the chromosome. And before the chromosome can attach back together, the broken piece does a 180-degree rotation. But this mutation doesn't usually cause any major issues as it doesn't change the number of bases. So the chromosomal strand is usually viable. But there are exceptions to this rule and it could cause some issues. But that gets into more complex genetics, so that will be it for inversion, and we'll be moving on to insertion.
Insertion is a type of mutation involving the addition of genetic material. An insertion of just one base pair could lead to a frame shift, which shifts the reading of the 3-base pair code, which can potentially lead to birth defects. However, if one or more 3-base pairs are inserted, it will not lead to a frame shift and is less harmful than the addition of just one base.
So the reason why adding 3 or a multiple of 3 base pairs is less harmful than adding any other number of base pairs is because there are some biological processes where DNA nucleotides are read in groups of 3. So these groups are called codons, and in the image above, you can see that each group of 3 nucleotides is labeled with codon 1, codon 2, codon 3, etc. And in the DNA strand at the bottom, the C and G nucleotides are removed, and this causes the fifth codon to be completely different from the fifth codon in the strand of DNA at the top. So everything after the fifth codon will also be different. And because of the shift in all of these nucleotides, this mutation can be called a frame shift mutation.
Now we will talk about deletion. Deletion is a type of gene mutation, put simply as something missing from the sequence. It can be just one nucleotide or an entire gene or even more. Much like as there's if suddenly part of the instructions on your Lego set missing, depending on what or how much is missing, the results can either be very big or a very small change. As you can see in the image, part of the chromosome is missing after deletion and leads to a frame shift, which essentially changes the way your body reads the DNA. We will not be able to tell what exactly this would lead to, as there are many results when it comes to mutations. That's it for deletion. Next, we move on to what these mutations cause.
Now we will be moving on to cystic fibrosis. Cystic fibrosis is a disorder that damages the lungs and digestive system by causing the production of abnormally thick mucus, which results in respiratory infection. You may be wondering, what causes cystic fibrosis? Cystic fibrosis is caused by a mutation in the CFTR gene. The CFTR gene stands for the cystic fibrosis transmembrane conductance regulator gene. This gene provides instruction for making a channel that transports chloride ions in and out of cells. The mutation in this gene would disrupt this function, preventing these channels from regulating the flow of chloride ions and water across cell membranes, which would cause thick mucus. Some symptoms of cystic fibrosis include wheezing, repeated lung infections, being unable to exercise, having a stuffy nose, poor weight gain and growth, severe constipation, and a cough that produces thick mucus. Treatment for cystic fibrosis may vary for each person, but doctors may recommend medications, mucus thinners to clear up the airway, or lung transplants to help fight the symptoms. That was all for cystic fibrosis. I hope you learned something new. We will now be moving on to sickle cell anemia.
Sickle cell disease is caused by mutations in the beta-globin HBB gene that leads to the production of an abnormal version of a subunit of hemoglobin, known as hemoglobin S. Hemoglobin is the protein responsible for carrying oxygen in red blood cells. Both the mother and father must pass on the defective form of the gene for a child to be affected. Otherwise, if only one parent passes the sickle cell gene to the child, that child will simply have the sickle cell trait. Normally, the flexible, round red blood cells move easily through blood vessels. However, in sickle cell anemia, the red blood cells are shaped like sickles or crescent moons. These rigid, sticky cells can get stuck in small blood vessels, which can slow or block blood flow and therefore oxygen to parts of the body. So as one could expect from this, various symptoms can appear in people, which usually show up at around 5 months. And the most common of them is anemia. Sickle cells break apart easily and die, leaving patients with too few red blood cells. Normal red blood cells usually live for about 120 days before they need to be replaced, but sickle cells die 10 times faster. They usually need to be replaced after 10 to 20 days. So that leaves the body in a shortage of red blood cells and therefore in a shortage of oxygen, which makes people very fatigued and has other numerous side effects in patients. We'll be now looking over Huntington's disease. Huntington's disease is an Huntington's disease is an inherited disease that causes progressive breakdown of neurons in the brain. Unlike some genetic disorders where both parents need to carry the mutated gene for their offspring to have the same condition, Huntington's disease can be passed down with just one parent having the mutated gene. Symptoms can develop at any time, but usually they develop in your 30s or 40s. Just as a note, if symptoms or the condition does develop before you're 20, it's called juvenile Huntington's disease. Huntington's disease impacts a person's movements, cognitive, and psychiatric abilities. Those with Huntington's disease usually live for only 15 to 20 years after the condition begins. Some symptoms of Huntington's disease are involuntary movements, difficulty speaking, difficulty organizing or prioritizing tasks, difficulty learning new information, insomnia, and social withdrawal. Next, let's move on to cancer.
So now we'll be talking about cancer. More than 50% of all cancers are caused by a mutation in a gene called P53. The p53 gene is very important because it is located in the cell nucleus and it plays a key role in controlling cell division and cell death. If the gene is damaged, it can't control the growth of cells, and this leads to cancer. Sometimes you have a higher chance of developing cancer if you have a family history of cancer. This is because the mutated genes associated with cancer can be passed down to you. Having a damaged gene that controls cell growth increases your chance of developing cancer. Some types of cancer that can be genetically inherited are breast cancer, colon cancer, prostate cancer, ovarian cancer, uterine cancer, melanoma, and pancreatic cancer. Both breast cancer and ovarian cancer are commonly caused by mutation in the BRCA1 and BRCA2 genes. If you have an increased risk of developing cancer, there are some ways to prevent it. For example, American actress Angelina Jolie found out through a genetic screening that she had a significantly increased risk of developing breast cancer due to a mutated BRCA1 gene and a family history of cancer. She decided to have a surgery called a mastectomy to reduce the risk of developing breast cancer. Her actions inspired other women to undergo this procedure and reduce their risk of developing breast cancer. We've previously presented about cancer, its symptoms, and its treatments. In addition, these factors vary depending on the type of cancer and how much it has developed. In general, cancer can be identified by a bump, which is usually formed by cells growing uncontrollably. There are also other types of symptoms for different cancers. Some common treatments include radiation and chemotherapy, but there are other types of treatments for different cancers. For example, we previously talked about CAR T-cell therapy, which is a biotechnological advancement in which a component of the immune system called T cells are genetically engineered to identify and attack cancer cells in the body. Next, we'll be moving on to other types of advancements in biotechnology that can help save lives.
To end off this presentation, let's talk about an advancement in biotechnology, CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. It is a technology tool that allows for the DNA of a genome, which is an organism's or cell's complete set of genes, to be rapidly modified in a highly specific way. CRISPR is a system that can disrupt DNA, causing gene inactivation. It can delete DNA and it can correct a gene or insert a new gene in a genome. CRISPR and the ability to edit DNA is helping to make major advancements for cancer research and treatment, as well as treatment and research for many diseases. CRISPR can allow the DNA to be manipulated so that the changes in the DNA that cause cancer or diseases can be corrected. Being able to edit DNA opens a whole world of possibilities for medical breakthroughs. Now for how it works. CRISPR consists of two parts, a guide to RNA and the Cas9 enzyme. In the lab, a small piece of RNA is created which binds to a sequence of DNA in a genome and to the Cas9 enzyme. The RNA recognizes the DNA sequence so that the Cas9 enzyme cuts the DNA at a specific location. Then once it's cut, the cell's own DNA repair machinery kicks in. For the disrupt action of CRISPR, a single cut will be made, prompting the addition or deletion of base pairs, leading to gene inactivation. As for deleting, two guide RNAs can target separate sites, and non-homologous end joining brings the ends together to delete the middle DNA. Lastly, for correcting or inserting, homology-directed repair will occur by adding a DNA template next to the CRISPR machinery. This might not make a lot of sense if you haven't taken biology yet, but all you need to know is that CRISPR is a system that allows changes to be made to DNA. To finish this up, let's go over some quick facts. First, CRISPR was actually developed using bacteria that had a natural DNA editing system. Also, aside from cancer, CRISPR has the potential to treat cystic fibrosis, hemophilia, sickle cell disease, and hepatitis B. However, before CRISPR can be used routinely in humans for the advancements it was made for, it will likely be many more years. Despite the benefits CRISPR may offer, there's lots of controversy over how ethical it is, since it is the actual editing of your genes, which could cause undesirable and unknown results, such as changes in future generations. That's all for advances made in biotechnology.
Okay, and as some people mentioned in the chat, we're going to be doing a Kahoot next to review some of the things that we talked about. And also, because I think in the last Kahoot, you guys mentioned that you guys liked playing the Kahoots. So you feel free to join from your devices. The join code is loading right about now. Okay, so the join code is 2888076. Okay, I think when we're just gonna wait for people to join and then we'll start at like 10:22 maybe. I think we have really fun nicknames every time we play. Okay, I think maybe we can get started right now. The game pin will always be at the bottom corner, so you can like keep joining if you haven't already. Okay, so the first question is, what is a point mutation? And then you have a change in a single nucleotide of DNA, a mutation that shifts the DNA sequence, a mutation at a few points in the DNA sequence, and a general term for all mutations. Okay, yeah, so most people got it right. It's a change in a single nucleotide of DNA. Okay. Why is it hard to play on a phone? I feel like it's a little bit easier because you can tap. Okay, so that's our leaderboard, and we'll just move on to the next question. So what is insertion? When one nucleotide is added to replace another, an addition of one nucleotide only, an addition of more than one nucleotide only, or an addition of one or more nucleotides. Okay, yeah, so it's an addition of one or more nucleotides, so you can add one or two or three or any number. Oh, on your phone, you have to switch between Kahoot and Zoom. I think if you play from two different devices, it might be easy. Okay, so there are some changes in the leaderboard. Okay, the next question is a true or false question. The environment affects changes in our DNA structure. Okay, so everyone answered true, and that's correct. So congrats. And then we have a small change in the leaderboard, and also Yeet has a streak of 3 correct answers in a row. Okay, the next question, what are the letters in the DNA sequence called? So you have adenosine, nucleotides, arrows, protein. Okay, so adenosine is like a name for one type of nucleotide, but in general, all the letters are called nucleotides. Okay, what happens during an inversion mutation? A broken piece of base rotates, a base goes missing, an extra base is added, or the chromosome strand turns upside down. Okay, so the correct answer is a broken piece of base rotates. I think this is a hard question. But so when a base goes missing or like is deleted, that's a deletion. And when an extra base is added, that's an insertion. So it sounds kind of similar to inversion. And the chromosome strand doesn't usually turn upside down because a mutation kind of refers to the bases in the nucleotides. So hopefully that helps a little bit. Okay. For insertion mutations, what is considered the least harmful? So you have an insertion of one base, an insertion of 3 base pairs, an insertion of DNA, or an insertion of genetic material. Okay, yeah, so the correct answer was an insertion of 3 base pairs, just because we mentioned like the codons and how they're like read in groups of 3. And then if you insert like one base pair, that shifts the whole sequence. And then DNA and genetic material are too broad a term to kind of describe this. Okay, so the leaderboard is staying the same. Next, we have a true or false question. Deletion only affects one nucleotide. Okay, so this is false because if you like delete a nucleotide, the nucleotides after it can shift, and that can cause like a frame shift mutation. And then the next question is, what causes cystic fibrosis? Damaged lungs and digestive system, lung transplants, thick mucus and respiratory infections, or a mutation in the CFTR gene. Okay, so cystic fibrosis is caused by a mutation in the CFTR gene. Thick mucus and respiratory infections are a symptom of cystic fibrosis. So it's not really like what it's caused by. And then damaged lungs and digestive system is also like an effect of it. And lung transplants are like a way to treat it. Okay, so now we have a true or false question. Mutations can form when DNA makes copying mistakes. So I think this refers to like when they make mistakes when they're copying like DNA to like replicate it. Okay, so this is true. when DNA is replicating itself, it needs to copy a lot of nucleotides, so they can like make mistakes sometimes, and that can cause mutations. Okay, the next question is a mutation in which protein causing the sickle cell anemia? So you have beta globin, hemoglobin S, alpha globin, or hemoglobin subunit beta. Okay, yeah, this is a really hard question. So it's actually caused by hemoglobin S, and the hemoglobin subunit beta, I believe, is like a subunit of the protein hemoglobin, but the specific type is called hemoglobin S. Okay, and then this is our leaderboard. Yeah, most people did get that one wrong, so don't feel bad if you got that wrong. The next question is, what ages do people usually develop symptoms for Huntington's disease? So you have as a child, as a teen, in their 30s or 40s, and then in their 50s or 60s. Okay, yeah, so the correct answer is in their 30s or 40s, and I think most people got this one right. And then thank you, Sarah, for putting that in the chat. The HBB option, which a lot of people chose, is the gene that mutates. It's all known as beta globin, and then this mutation causes the production of hemoglobin S. So they're somewhat related. Okay, and then we have Greenfish in 5th place, so that's really good. Okay, the next question, mutations in which gene most commonly lead to cancer? So you have the BRCA1 gene, the CHEK2, P55, 53, and then PALB2. Okay, so the correct answer is the P53. More than 50% of all cancers are caused by this gene. I think maybe many of you chose the BRCA1 gene because we talked about Angelina Jolie. But this one, it's also common, but it's for breast cancer and ovarian cancer, so it's not applied to all cancers. So in general, a mutation in the P53 gene would most commonly lead to cancer. Okay, so the next question, which type of cancer is not genetically inherited? You have breast cancer, colon cancer, melanoma, and then lymphoma. Okay, yeah, so the correct answer is lymphoma. We had like a list on the slide about cancer, and then in the list, it didn't include lymphoma. And then most of you got it right, so congrats. Okay, so next we have a true or false question. Point mutations almost always cause tremendous change in the underlying organism. Is that true or false? Okay, so then we got like a 50-50. This is a false because point mutations are like small mutations, like substitution or something like that. So it doesn't like cause like a frame shift mutation. So that's why it's like it doesn't cause like tremendous changes. It might cause like a small change, and then it can like sometimes cause like a tremendous change, but like it's not common. Yeah. Okay, so for our last question, we have what can CRISPR not do? So we talked about what it can do, but then what can it not do? So your options are delete DNA, switch DNA, insert DNA, or correct DNA. Okay, so CRISPR can delete DNA and then insert another section of DNA to correct it, but it can't like switch it. So that one was the correct answer. Okay, and now we'll like look at the podium. So in third place with 11 out of 15, we have Yeet. And then in second place with 10 out of 15, we have Sora. In first place with 13 out of 15, we have Marin. And then our runner-ups are Frijoles and Riley. So congratulations to the winners. And thank you everybody who played. Yeah, it was kind of difficult to play in like on like the same device. But yeah, you guys did a really good job. Okay. We'll go back to this slide right here. So like after playing the Kahoot and after the presentation, did you guys have any questions? You can feel free to put them in the chat. I know this is like a weird topic because some of you haven't taken biology yet, but I think it'll be a good a good introduction for your biology courses in high school or like in college. And if you've already taken biology, I think it's a good way to review. Patrick, did you have a question? Okay, well, you can type it in the chat. And then if you guys don't have any questions, I know like school is starting for a lot of you. And so we want to wish you all good luck, especially if you'll be returning in person. I think it'll be great to see all of your friends. And then our next session for the fall semester, that will start on Sunday, September 12th at the same time. And this is just so you guys can get used to like going back to school, going to like high school or middle school, or wherever you're going. Oh, why did they make it so weird to name? Oh, for the proteins. I think it's just like a scientific name. So like if you're a scientist, right? Maybe you don't think it's weird. But yeah, like Zachary said, just to like abbreviate, it probably like stands for a scientific name and it's just like an abbreviation to make it shorter and easier to say. But yeah, that's all we had. Oh, yeah, Sonia said they're a way of classifying and determining different genes, and so it's easier to remember than the long scientific names. Yeah, so like an abbreviation. Yeah. Okay. So that's all we had planned for today. And I think we have like 1 or 2 minutes left. So you guys can feel free to leave if you want to. Next semester we'll be talking about physiology and anatomy, but we'll be sending out more details about that in the recap emails. Yeah, so thank you so much for coming, everyone. I'll stop the recording.